Good evening, students. Welcome back to my class. Today, let us discuss about gastrulation. After the implantation of the blastocyst into the maternal uterine endometrial lining, gastrulation begins. That is the formation of three germinal layers: ectoderm, endoderm, and mesoderm. The inner cell mass, or the embryonal knob, which is present at the animal pole of the blastocyst, undergoes certain changes. That is, they show characteristic movements. This characteristic movement is known as morphogenetic movements. Now let us discuss about the process of formation of these three germinal layers and the extra-embryonic membrane. This is the structure of a blastocyst. We can see trophoblast layer, inner cell mass, and blastocell. At the beginning of the gastrulation, the cells which are present at the at the free surface of the inner cell mass undergo differentiation, that is, they flattens. This layer is the first endodermal layer and it is known as hypoblast. The hypoblast layer then undergo division, rapid division. The cells then move or extend downward, lining the inner surface of the trophoblast. The newly formed lining is known as exocoelomic membrane, and the blastocell now have a new lining, and thus it is converted into primary yolk sac. Not only the free surface cells undergo differentiation, but also the other cells of the inner cell mass undergo differentiation. Here, the cell becomes columnar. These forms the ectodermal cells or the epiblast. The epiblast is now known as embryonal disc. It just looked like a disc. We can see the structure here. This is the embryonal disc which give rise to the embryo, and this is the endodermal layer. Now, the embryonic disc is divided into three parts. This part, it is known as cephalic margin, which give rise to the head, embryonic disc, and the caudal margin. Next, a space is formed in between the ectoderm or epiblast and the trophoblast. The space which is formed, or the cavity which is formed, is known as amniotic cavity. The roof of the amniotic cavity is lined by amniogenic cells which is derived from the trophoblast. Now, the trophoblast cells also undergo rapid division, thus to form a new layer of cells which is known as extraembryonic mesoderm. After some time, certain small vesicles arise in the extraembryonic mesoderm. These vesicles unite to form a cavity which is known as extraembryonic coelom. The extraembryonic coelom now divides the extraembryonic mesoderm into two different parts or layers. One lining the trophoblast is known as somatic layer, and the one lining the secondary yolk sac, amnion as visceral layer. Now, this is a region at the animal pole which is not having the cavity or the coelom. It is known as the connecting stalk. This connecting stalk now shifts towards the caudal margin. As we can see here, the connecting stalk was at the animal pole. Now it has shifted towards at the end of the caudal region of the embryonic disc. Here it is shifted because the nutrition will be easily absorbed by the embryonic disc. Next, let us see how the three germinal layers are formed. This is the embryonic disc, as you can see here. Now this is the embryonic disc. This is the caudal end. A thickened structure forms along the middle in the epiblast near the caudal end. This is known as primitive streak. Here we can see primitive streak. At the cranial end of the embryonic disc, the primitive streak expands to create a primitive node which contains a circular depression called primitive pit. This depression continues along the midline of the epiblast towards the caudal end of the streak. This is known as primitive groove. Once formed, cells of the epiblast migrate inward towards the primitive streak. Here we can see inward movement of the cells of the epiblast. As a result, the epiblast at this part detaches from the endodermal layer. Now these cells completely replaces this endodermal layer to form a new endoderm layer. Some more cells keep on entering and form a layer of cells in between the endoderm and the epiblast. This is known as mesoderm. The remaining cells of the epiblast forms the ectoderm. Hence, three layers of cells are formed: ectoderm, mesoderm, and endoderm. Ectoderm, mesoderm, and endoderm further give rise to different parts of the body. Thank you.